viewers and learners of the PG Diploma in Environmental and Occupational Health. I introduce myself as Dr. Susmitha Bhaskar, working in the School of Interdisciplinary and Transdisciplinary Studies, Indira Gandhi National Open University. Today we will discuss the unit Mutations and Mutagens. The course code for this course is MEV 004. The learning objectives of this module includes definitions and concepts related to mutations, mutagenic agents, mutagenesis and so on. We will also explain and understand the various types of the mutagenic agents and the toxic reactions and how they form certain toxic effects. We will also see the various kind of the genetic abnormalities, the toxic chromosomal aberrations and the various agents, for example whether it is radiation, whether there are certain chemical agents, all those agents that are bringing about the mutagenic effects. And finally we will discuss these with the help of the case studies and the environmental factors that are responsible for bringing about mutagenesis. Mutations. So mutation is a heritable change in the genetic material. So here you see the two diagrams of the double helix strand of a DNA. You will see before and after a mutation has occurred. So before a mutation has occurred, you will see that those base pairs and how it aligns with each other and the helix appears very normal. Now after some mutation, it could be due to some chemical agent or it could be due to some radiative action, you can see that those base pairs are misplaced and there is that double helix structure is not normal, it is disrupted. The importance of such studies. These are very important to geneticists and genetic variants, they possess mutant alleles of the genes. So genetic variations will fuel the evolutionary change. So these are very important factors. So genetics plays a very important role in the evolution which is fueling the evolutionary changes, whether it is from the plant species, in the animal species and also in the human evolutionary process. Hereditary mutations. So these are inherited from the parent and they are present throughout life in every cell in the body. So sometimes we will see that the child has a natural intellect and natural interest in music because maybe the musical genes or the artistic genes are coming from the parents or from the grandparents. So there is a lineage, there is some gene which is responsible for bringing about this. Now this is a positive aspect. There could also be certain other problems, for example something which is related to a deficiency in the body or maybe diabetic gene or some other diseases which are also inherited through the genes. So a parent may have a color blindness, so then the child may also have the color blindness problem, or the grandparent may have a hypotrichosis where tuft of hairs appear on the ears. Now this could be a Y chromosome inherited and it could travel in the generations. So hereditary mutations, they could be inherited from the parent and they are present throughout life in every cell in the body. The germline mutations, so they are in the germline, it is carried through the parent's egg or the sperm cells, and during fertilization the DNA is received from both the parents. So whether it is the X chromosome is coming from the either from the mother or from the father, and the Y chromosome is coming from the father, from the sperm. So therefore when both unite, you find XY which is a male fetus, and you can find XX which is a female fetus or the embryo which is developed. So if this DNA either in the X or in the Y, if there is a DNA mutation, if there is a change, a hereditary change, then the child will have the mutation in his or in her cells. So this is what we say by saying that it is inherited from the parent. It is a hereditary mutation and it will remain for life in the cells of the body. Somatic mutation. So this is not inherited from our parent. It can occur at some time during a person's life. It is present only in some certain cells, not in every cell of the body. It is caused due to environmental factors, UV radiation from the sun, due to errors when the DNA copies itself during cell division process, and this somatic mutation will not be transferred to the next generation. So sometimes we have this feeling that you have some problem, and whether the child will also inherit that, and you go to the doctor and you go for counseling. So it depends on the mutation type, whether it is somatic or whether it is hereditary in nature, and whether the child will inherit that or not. Now these are clearly more explained with the help of certain diagrams where there is a germline mutation and the somatic mutation. Now in the germline mutation, where you find the sperm and the egg, so anything which has occurred, any damage which has occurred or a mutation which has occurred in the chromosomes, in the Y or the X chromosome of the parent, that can then be directly, it will come to the child, and the entire organism will carry this mutation, and then half of the gametes will also carry those mutation which will be transferred to the next generation. Somatic mutation. Now here it could occur at any part, it is not coming from the parents. At any time of the individual, at any part, it could be either in your hand, in your arm, or it could affect your legs, anywhere a mutation can occur, and it is not necessarily that the gametes and the gametes will carry this mutation and that it will be transferred to the next generation. So this figure is clearly showing you the parental gamete type, the embryo, whether the embryo will be affected by the mutation, the organism when we grow up, whether we will have it, and whether we are capable of transferring that mutation to the next generation. So to function correctly, each cell depends on thousands of proteins to do their job effectively. Gene mutation prevents one or more of these proteins from working properly. By changing a gene's instruction for making a protein, a mutation can cause the protein to malfunction or to be missing entirely. Now in this figure, you can see the DNA code for an amino acid sequence. In the top you see the DNA bases, that is cytosine, the adenosine and the thymine, whether they are coding for the amino acid. And when the nucleotide is replaced, now on the top we had CAT, and here you find the CAT the sequence being interrupted, and a different nucleotide is replaced, that is the adenine A is replaced with C, cytosine. So this will code entirely for a different protein. So therefore incorrect amino acids will be replaced when the coding is altered, and the altered protein will actually code for a different amino acid. When a mutation alters a protein that plays a critical role in the body, then it can also disrupt normal development. For example, phenylketonuria. So this is a condition which is caused by mutations in one or more genes, and it is called a genetic disorder. So here you see whether there is mutation or whether there is no mutation, whether the phenylpyruvic acid, that is there is a mutation which has occurred, and you can have this phenylketonuria problem, or whether the normal functioning will be something different. So mutations can alter the normal development, and it can entirely disrupt even the hormonal and the protein synthesis in the body. Mutagens. So this can cause changes to the DNA, and they are genotoxic. They can affect the transcription, replication of the DNA that can lead to cell death. Deleterious mutation can result in aberrant and impaired function for a particular gene. Mutagens may also be carcinogens. Some mutagens, they can exert the mutagenic effects through their metabolites. They become carcinogenic. They are dependent on the metabolism of an organism also. Different mutagens act on the DNA differently. There are powerful mutagens which can result in chromosomal instability. They can cause chromosomal breakages, translocation, deletion and inversion. Such mutagens are also called as clastogens. Now the changes in the structure of the chromosome. So here we can find that they can cause translocations, deletions, duplications, inversions, isochromosomes, dicentric chromosome and ring chromosomes. The first is translocation. This occurs when a piece of one chromosome breaks off and it is attached to another chromosome. So here I have explained it with a figure. We have chromosome A and chromosome B depicted in the violet and the pink colors, and you will see that in the violet chromosome A, one part of the chromosome is breaking off and this is getting inserted along with the chromosome B. So you have a different chromosome, and this is an effect, a translocation effect. Deletion. This is occurring when a chromosome breaks off and some genetic material is totally lost from that. So duplication, when one part of a chromosome is duplicated many times. So in the top figure you can see deletion. Okay, I have given that with the help of certain colors: green, orange, blue and violet. So in this you see the blue part of that chromosome is deleted. So the new chromosome which you see there, that part of that chromosome is lost and the genetic material is thereby lost. In the second part, which is duplication, you see that the yellow part of that chromosome is duplicated twice. So you see that that part of that genetic material is duplicated many times. Inversion and the breakage of a chromosome can occur in two places, and the resulting piece of the DNA will be reversed and it is reinserted into the chromosome. What happens as a result? The genetic material may be lost, may not be lost as a result of the chromosome breaks. So here you see there is the numbering which is going from A, B, C, D up to K, and down part of the figure you can see it is ABC, and that one part of that green figure which is cut and it is being reversed and reinserted into that part. Okay, it is getting reinserted as HGFED. So it is possible that one part or so can be lost, or some part of that material, genetic material may be lost or may not be lost. So this is a problem with the reinversion, where it is reversed and inserted in the process. Isochromosomes. These are the chromosomes with two identical arms. Either they have two long arms or two short arms. So right now when I am explaining this, you may not be very clear with why we are explaining this, but in the later part of the slides, the later part of my lecture, you will understand when we have the Turner syndrome, we have the Down syndrome, so many of the mental retardation which is occurring among the people, among the children that are born. It is occurring due to the effects of these mutagenic agents from environmental factors, because it is occurring at the DNA level and mutations are occurring, and that is why the children are getting the cleft palate and certain other abnormalities. So in this, coming back to the isochromosome. So here you see that two of the chromosomes, okay, either they could have the two short arms or they can have the two long arms. So that is the isochromosome. Ring chromosome. This is when a chromosome is breaking into two places. You know, here it is breaking on the top and also at the bottom path, and then that will fuse together to form a ring or a circular structure. Here again the genetic material can be lost. So therefore, now we have discussed about the mutations. So chromosome numbers, do they affect the health and the development? The human cells, they contain 23 pairs of chromosomes and total of 46 chromosomes in each cell. The change in the number of chromosomes can cause problems in the growth of the cell, in the development and also function of the fetus that is being formed. There can also be gain or loss of chromosomes from the normal 46, and that can also be called as aneuploidy. Trisomy, this I think people are very familiar with. This is a presence of an extra chromosome in cells. They can have 3 copies of a particular chromosome instead of the normal 2 copies, because we carry the 2 copies with us when we have 3. So that is the trisomy 21. Down syndrome is an example, where instead of 2 we have the 3 chromosomes over here. So here, this is the figure that is showing in the chromosome 21, you will see that there is one extra chromosome. That is why it is called as trisomy 21. Tri means 3. So 3 copies of the chromosome in 21 in each cell, for a total of 47 chromosomes per cell instead of the normal 46. This is how a Down syndrome child appears. They have a mongoloid appearance. There is flat nose of the face, and the eyes, they are actually distended. I mean, they are wide apart from each other. There is a single palmar crease in the hand, and in the feet they can have, there is a gap between the toe and the other fingers. There can also be more creases, and the children could have mental retardation. Monosomy. This is a loss of one chromosome in the cells, and it is another kind of aneuploidy. They can have one copy of a particular chromosome in the cell instead of two copies. Turner syndrome is a condition that is caused by monosomy, because there is one less. Instead of two, they have only one copy of that particular chromosome. So here, this you are seeing here in this chromosome, it is instead of XX, it is XO. So one chromosome is lost over here. So women with the Turner syndrome, they have only one copy of that X chromosome in every cell, for a total of 45 chromosomes per cell instead of 46. So can these changes in the mitochondrial DNA affect the health and development? Mitochondria, they have a small amount of DNA known as the mitochondrial DNA. Inherited changes in the mitochondrial DNA that can cause problems with growth, development and function. They can disrupt the mitochondria's ability to generate the energy efficiency for the cell. Effects. This can be seen on the heart, on the brain, muscles. It can cause diabetes, kidney failure, dementia, hearing loss and also abnormalities involving eyes and vision. Mitochondrial DNA is also prone to somatic mutations like Alzheimer's and the Parkinson's disease. Multifactorial disorders. We have the sickle cell disease and cystic fibrosis. These are caused by mutations in a single gene. Type 2 diabetes and obesity. They do not have a single genetic cause, but they have multifactorial disorders. Specific factors that cause most of these disorders are still under study. Radiation. So this could be due to the UV radiation, X-rays, by the benzoyl peroxide which is applied on the face. Then there could be due to the Helicobacter pylori, that is a bacterial agents which are infectious agents. It could be due to the HPV, there is a human papilloma virus, cigarette smoke and so on. Also certain barbecuing agents and also nitrate and nitrate preservatives. All these can bring about the mutagenic effects. In this flow chart, you can see that the mutagens can be classified into physical, chemical and biological agents. In the physical, we have radiation and heat. In the chemical, we have the base analogues, intercalating agents, alkylating agents, deaminating agents and metals. In biological, we have the transposon, we have viruses and we have bacterial agents. Radiation. This was the first mutagenic agent known, and its effects were even reported as early as 1920s. So ionizing radiation that can react with DNA and other biological molecules, and it is a very important mutagen. This can cause burns, skin damage, and it can increase the risk of mutation as well as cancer. The type of radiation, so like for example X-rays, that can induce the mutations forming free radicals and it can ions, and it can bring about addition, deletion, transitions and transversions. Now that you have understood about these additions and deletions which I was explaining, so now you know how the X-rays can bring about the mutations, that is their mode of action. Same way gamma rays, alpha and beta particles, they can bring about chromosomal gene aberrations. Fast and thermal neutrons and UV rays, they can bring about chromosomal breakage and the gene mutations. Exposure to the radiations. In the Hiroshima and the Nagasaki atomic bombing in 1945, several of the people were exposed to this harmful radiations, and the survivors, they had these mutagenic effects due to this radioactive output. Now some of the factory girls who were affected and killed by the radium, because they were painting these radium paint on the watches and the clock, and this radium was actually very hazardous, and without knowing, because they were actually using a small paintbrush for painting. So it could even, it would have like they had applied it on their cheeks, because it was seen that this also gave a good glow on their cheeks. It was also used in some kind of cosmetics. So they applied it, and even unknowingly also the brushes used to strike their face and their skin. And then later on in 1925, scientists discovered that radium was hazardous, and this was the component which was deposited in the women's bones, because most of these girls, they saw that their legs were trembling, and they had the arthralgia problems, then their teeth started falling off, and then their hairs also were getting affected in the process. So then they realized that this radium was deposited in the bones, and that was a bone toxicity effect which was causing the teeth and everything to fall off. Marie Curie, she was a scientist, and she was working on these radioactive emissions and radiations for several of years, the noted Nobel laureate, and she herself died due to leukemia and overexposure to the radioactive emissions. The chemical mutagens are also there, and the first mutagenic action of the chemical was noted as early as 1942 by Charlotte Auerbach. He showed that the nitrogen mustard was a component of poisonous mustard gas which was used in the World War I and II, and they could cause mutations in the cells. Mustard gas. So they are weapons that were used by the Germans in 1917. The soldiers were incapacitated when this mustard gas was being applied. There is severe swelling of the eyelids, there is skin reddening, blisters to the throat, chest and nausea. All this occurred among the people. And it was a terror actually that was faced during the World War I that led to the ban of this mustard gas. In the 1925 Geneva Protocol, they banned the use of this chemical. Then there are a group of chemicals, that is the alkylating agents, which consists of ethyl methane, sulfonate, the ethyl ethane, then ethyl ethylene amines and so on. They bring about the ATGC transitions and also certain other transitions which are causing their mode of action. Base analogues, the acridine dyes and others like the nitrous acid, sodium azide, hydroxylamine, all this can bring about the mutagenic effects, and their mode of action could be by deletion, by additions, frame shifts and also transitions. Benzopyrene and other polynuclear aromatic hydrocarbons. Beef which is grilled over the charcoal fire, they can contain these polyaromatic hydrocarbons. The source is the smoke that is generated when these spiralized fat dripped from the meat onto the hot coals. Thus meat with the highest fat content, they acquire the highest levels of this PAH. It is also found in the smoked food, and also roasted coffee can be one source of the PAH. Benzene. They are industrial solvents and precursors in the production of drugs, plastics and synthetic rubber and the dyes. Metals such as silver, cadmium, chromium, copper, mercury, molybdenum and lead. In the same way, arsenic, cadmium, chromium, nickel and their compounds also have been shown to have mutagenic effects. Aflatoxins and mycotoxins, which have from the fungal source, they can also cause this problems. Herpes virus, they can also bring about the problems, and transposons can also bring about the DNA mutations. Therefore, the summary from this lecture, we can conclude that mutations, we have seen the several types of the mutations. They can be induced from several agents by radiations, that is or heat, that is a physical, the chemical and the biological agents. There could be several types that are causing them. The several types of mutations we have discussed, whether it is additions, the deletions, the transitions and so on. We have also seen the several mutagenic agents, and we have discussed it with the help of certain case studies. So protection is, we know our environment, the factors that are responsible from the environment which are causing such mutagenic effects, especially those mothers who are expecting their children should be very careful about what they are consuming, what they are eating, and the radiations that they are exposed to, also their workplace, even us also. And because of both the gametes, the hereditary mutations which are carried in the germ cells, so both the male and the females have to be cautious about their workplace and the type of occupation that they are being exposed to at several times in their life cycle. And we can also protect ourselves from taking antioxidants by consuming the cruciferous vegetables and resveratrol, which is also obtained from good red wine. Thank you for your patient listening.